Welcome back. Today let's discuss microscopy. Microscopy is a fascinating and essential field in science that allows us to observe objects that are too small to be seen by our naked eye. In cell biology, it is a vital tool as virtually everything we study in cell biology is far too small to see with our naked eye. The first and oldest microscopes were developed about 400 years ago and used visible light from the sun, with glass lenses to magnify the sample. In the last 100 years, there have been some pretty significant advances in microscopy. About 80 to 100 years ago, we discovered that using electrons to image samples greatly increased both the magnification and resolution of the microscope. In the last 50 years, laser diode microscopes have been made. The choice of microscope depends on the material, the process, the resolution, and the depth of the sample. Before moving on, let us revisit the physics behind how microscopes work. Let's recollect the basic principles of the wave and particle properties of visible light. Electrons also have both wave and particle-like properties, so many of the principles of electron microscopy are similar to those of light microscopy. All microscopes have limitations on the size and detail with which they can be used to visualize objects based on the characteristics of the particle being used to create the image, whether photons or electrons. The wavelength at which these particles travel has a direct impact on how much detail can be seen in the image generated. Let's look at some common terms in microscopy. Magnification. The process of enlarging an object to examine its details. This is done using lenses that bend light or electrons to enlarge the image. In other words, the magnification of an image is simply how much we have enlarged the object in order to observe it. Resolution. The ability of a microscope to distinguish two points as separate, or we can say it as the closest spacing of two lines that can be distinguished as separate entities by a microscope. Higher resolution provides a clearer, more detailed image. The resolution of any microscope is limited by the wavelength of the particle being used to illuminate it. Contrast. Contrast is the difference in light intensity between an object and its background. In microscopy, improving contrast helps differentiate between various components of a specimen, such as cell structures. Staining and using special techniques can improve contrast. Numerical aperture, or NA, is an important concept in microscopy. The numerical aperture of a lens measures its ability to gather light and resolve fine specimen detail at a fixed object distance. A higher numerical aperture allows for a clearer and brighter image. Field of view is another key term to understand. The field of view refers to the visible area seen through the eyepiece. As magnification increases, the field of view decreases. This means you can see less of the object at higher magnifications, but with more detail. Depth of field is the thickness of the specimen that is in focus. At higher magnifications, the depth of field is smaller, meaning only a thin slice of the specimen is in focus at a time. This is why thicker specimens may require focusing at multiple layers to obtain a complete image. Illumination is crucial in microscopy. Proper illumination ensures that you get the best possible image. Light microscopes typically use visible light, but special types, like fluorescence microscopes, use ultraviolet light. The light source needs to be directed properly, and the intensity can be adjusted for optimal imaging. That's all for today. Let's jump into different types of microscopes in the next class. If you're enjoying the content and want to learn more, don't forget to like, share, and subscribe to get access to more amazing lessons from me. Thank you.